Okay, so this is the beginning of our notes for biology, ninth grade. You can go ahead and title your notes Unit 1, Experimental Design. I will collect notes at the end of each unit, basically a binder check. Now it's up to you if you want to take notes traditionally, just pen and paper, writing in your journals. If you'd rather take notes digitally, like either using screen and screen or watching this video on a cell phone and taking notes on the computer, that's okay as well. I prefer writing myself, but whatever way you can stay organized with your notes is okay with me. But regardless, go ahead and title Unit 1 Experimental Design. Now, before you get going, I do color code my notes with the red and black system. Anything that's in red, I want you to read it and I want you to understand it, but it's not necessarily something that you have to write down. Um, Now, it still means that if you see something in red that you don't understand, you want to mark it so you can ask a question about it during the next class. It doesn't mean that you should skip that stuff. It just means that whatever I write in black, that's what I consider to be the main points, the main ideas, the stuff that I want to make sure that you know. Further, try to get out of the habit of simply writing down word for word what I have on the screen. You definitely want to read, digest, and then try to simplify and put things down in your own words. All right, so we don't need to write this down, but I think you should know that science is the study of the natural world, and that happens through observation and experiment. And that's not just biology, that's any type of science. Now, experimental design, which is the main avenue that science has performed, it's the standard method that relies on controlling experimental variables in order to make comparisons between results. And the key difference there between just observation and experimental design is the idea of controlling different variables. When we control variables, we have a baseline to try to figure out which variables are the ones that are responsible for whatever change we may observe. That'll make more sense as I give you some examples. All right, so to put these steps in order, the first thing that occurs during experimental design is observation. I'm going to walk you guys through a hypothetical scenario, which is this. You notice that there is green mold growing in the corner of your bedroom. So that is your step one observation. Now, once you make that observation, you come up with a question or a problem. Take a second and ask, what question would you come up with if there was green mold growing in one corner of your bedroom? Probably something like this. Why is the green mold growing here? And then maybe a little further, how come it's just growing in that one corner of my bedroom and nowhere else? Now you come with a hypothesis. A hypothesis is an educated guess based on those observations, something that I'm sure you guys already know. Um, an example of this, oh, sorry, we put this often, not always, but often in a if-then statement. This would be an example of the if-then statement. If I leave wet clothes in the corner of my room, then mold would grow. So maybe the hypothesis is based on the idea that moisture leads to mold growth. If one corner of the room is always dark, that will lead to mold growth. That would be a hypothesis based on the idea that maybe sunlight kills mold, so darkness allows mold to grow. Um, Once you've come up with a hypothesis, you then run a controlled experiment. So you test your hypothesis by designing a lab or an experiment. You run a procedure, you gather information. We call information data in science. That data can be numbers, it can be pictures, it can be descriptions. There's all sorts of data. But regardless, that information you gather is meant to try to answer your question. So here's the example experiment that I would set up for this lab. I find a different room without any mold in it, and I create 4 experimental groups in each of the corners. By doing this, it allows me to control which variables may or may not be impacting the mold growth. and basically test out those hypotheses one at a time. So the first corner is going to be my control group. The control group does not have any experimental variables. The control group is basically used as a comparison to those experimental groups. So the first corner is light, it's dry, there's nothing in it. Now the second corner is just like the first, it's dry and it's empty, but there's no light there. So that is, my first experimental group is testing the possibility that darkness may lead to mold. Now the third corner is light and it's dry, but there's food in it, testing to see if food leads to mold growth. And then the fourth corner is light and there's no food in it, but there's wet clothes or damp clothes in it to see if dampness or moisture leads to mold growth. Now, a question that I want you to be able to answer here, why can't I put food and wet clothes in the same corner? Take 30 seconds, pause the video, and try to answer this before I give you the correct answer. Why can't I just save time and test two different things in one of the corners? Hopefully you're able to come up with the correct response, which is if you don't isolate the variables, meaning if you don't test one at a time, then you actually don't know which variable caused the change. For example, if mold grows in a place where I put food and wet clothes, that doesn't help me at all because I don't know what caused the mold. Maybe it was the food, maybe it was the wet clothes, or maybe it was a combination of the both. So it's really important that we always isolate variables, and that's the theme that will come up throughout the unit. Okay, after we run that experiment, we get to the results. It's important to recognize that results include what happened, but not necessarily why it happened. So here's my example results. I run this experiment for 14 days. Corners 1 through 3 do not have mold in it, whereas corner 4, where I put the wet clothes, does grow mold. So that leads me to a conclusion. Why? Now it's important to recognize that this step does not prove anything. It simply suggests a likely possibility, but we always want to stay away from the word prove in biology. And an example here would be based on the experiment, dampness or moisture causes mold. Darkness does not, food crumbs do not, but it's actually the dampness that causes the mold. And then we repeat the experiment, and potentially that can form a theory. If enough scientists repeat and agree upon results, that leads to many of the scientific theories that we have today. If the hypothesis is not supported, that's fine. It's always okay to be incorrect when we're making a hypothesis. We just need to change it, revise it, start again. But if it is supported and repeated and repeated and repeated enough times, it can be turned into a theory, something that is widely accepted. An example theory here would be that dampness leads to mold growth. It is a valid theory that mold requires dampness in order to grow. Now, within that experiment, that hypothetical experiment we just ran, there's two types of variables, and it's important that we understand the differences between these variables and how they're used. So the independent variable is what the scientist changes within the experimental group, whereas the dependent variable is some sort of change that is caused by the independent variable. An easy way to remember this is the independent variable is what the scientist manipulates, and the dependent variable is what the scientist measures at the end of the experiment. So if you think back to the example I just gave you, the independent variable is pretty straightforward. You had 4 corners. What did the scientist manipulate? The scientist manipulated what was put in the corners. So the independent variables were darkness and dampness and food, whatever was put into the corners. What was the dependent variable? Well, you need to ask yourself, what was measured at the end of the experiment? The scientist measured mold, or the absence or presence of mold. So that means that the dependent variable is the growth of mold. Further, there's two types of groups that we need to establish when doing experimental design. Experimental groups are what has the independent variable applied, whereas the control group does not have the independent variable applied. It's something that's used to compare with the experimental groups. So again, going backwards to our example experiment, the experimental groups were corners 2, 3, and 4. Those were all given some sort of variable, whereas the control group was corner 1 because the corner number 1 had no had no independent variable applied to it. Okay, looking into characteristics of good experimental design now. Some of these are a bit redundant from what we just talked about. Some might be obvious, some might be not so obvious. So it should have a hypothesis. Again, that's not a rule in science, but it's good practice, especially where we're at now, that we do some research and we come up with an educated guess of what we think is going to happen before we run the experiment. It should be reproducible. That part's really important, that scientists are able to replicate and reproduce other scientists' work. If you do an experiment and find something really cool, but no one else can replicate what you did, it's really not that valuable to the world of science. So that's where writing a procedure comes into play. By writing a really detailed procedure that can be replicated step by step, we include the amounts and the measurements and everything that we did. It allows other scientists to copy our work, and that in turn leads to the idea that science can be reproducible. The experimental group has the independent variable applied to it. We already mentioned that. And then there's a control group that does not have the independent variable, and this is used for comparison. This is a big one. Please put a star next to number 5. Highlight it, circle it, underline it. You need to try to keep all unwanted variables constant. What I mean by this, think back to our example experiment. What if half of your room was really, really warm and the other half of your room was really, really cold? Would that be a problem? Absolutely. It would be a major problem because it means we're no longer isolating variables. Instead of testing dampness, we might be testing dampness and coldness, or dampness and heat. What if a corner of your room is where your pet dog always hung out? Would that be a problem? Absolutely, because that is not part of the experiment, so we want the corners to be the same temperature. We don't want a dog in any of the corners. We don't want anything in there that could potentially change our data. There should be multiple trials. So again, if you do this experiment and you see that the wet clothes grew mold, that doesn't prove anything. It suggests that dampness might grow mold. And it's really important that you then do it again and again to see if you get the results. Now, if you do that experiment 5 times and all 5 times you see the same data, then that improves the reliability. That makes it much more likely that your results happened for some sort of scientific reason, not just because of chance. Speaking of chance, we want large sample sizes. So again, if you had 1 bedroom, that's okay. But if you had 10 bedrooms, which I don't think any of us have, but if you had 10 bedrooms, that would be a better experiment because it's going to reduce the chances of luck. And then lastly, your data and results should be both qualitative and quantitative. Qualitative is descriptions, such as the mold was green, the mold was fuzzy, the mold smelled like peanut butter. Quantitative is anything dealing with numbers. The mold grew 14 centimeters high. When I scraped off the mold, it weighed 9 grams. Those would be quantitative values. And then last but not least, all conclusions are tentative. The word tentative means subject to change. So basically, when I just said we don't want to ever say prove in biology, it's because biological results of any experiment results are tentative. Other scientists, other discoveries might occur that change our future body of knowledge, meaning what we know now to be true could potentially change in the future. That's one of the most interesting parts of biology, in my opinion. Okay, so to wrap up these notes, you're actually done writing for now, but I want you to just get a little bit of practice of what is wrong with the following experiments. So I make a hypothesis that putting Skittles in the soil will make a plant grow really tall. I put Skittles in the soil when the plant was 3 inches tall, and then 2 weeks later it was 10 inches tall. I conclude Skittles make a plant grow tall. Pause the video and try to brainstorm what is wrong with this experiment. There's no control group. I should have had another plant that I added 0 Skittles to, because there's a chance that that plant would have grown from 3 to 10 inches, whether it had Skittles or not. So without a control group, I actually don't know if the independent variable actually affected the dependent variable. So without a control group, that's an invalid experiment. Okay, let's try it again. Same hypothesis. I plant 2 seedlings under a tree. One under a tree and one in an open field. I place Skittles in the soil of the plant in the open field, and I use the plant under the tree as my control. After 2 weeks, the Skittles plant grew taller than the control plant. I conclude Skittles makes plants grow tall. Pause the video. Brainstorm. What is wrong with this experiment? Okay, we have a control group this time, but there are no constants. The main lack of constants is the fact that the plant under the tree and the plant in the open field are exposed to different elements. In the field, there's probably more sunlight, there's probably more water, less competition for resources, where under the tree is probably not a great place for a plant to grow. So even though I had a control group, there was a lack of constants in that experiment. Okay, here's the way we should do it. Hypothesis. I put 4 plants of the same species under the same conditions. I have one plant that receives no Skittles. That's my control group. The other plants, I put some different number of Skittles to maybe test out the different variations. All the plants grow to be the same height. I conclude Skittles have no effect on plant growth. This experiment is valid. Maybe not the most interesting experiment ever, but it scientifically, in terms of those rules of experimental design that I went over, this experiment is valid. Okay, so this ends the first half of your notes on experimental design. So we can stop here for now, and the second half of this video will be on collecting data and using graphs. Okay, so why do scientists use graphs? Really for 2 reasons. Number one, they show data numerically. Think about what's easier to understand, a big list of numbers or turning those numbers into a picture. A picture is much easier to understand. And by turning it into that picture, that visual, it allows us to show off patterns and trends, and allows us to make sense of that data. So there's 4 main graphs that scientists tend to use. You can take a second and try to think of what those 4 are. My guess is most of you were able to think of 3 of them, bar graph, line graph, pie chart. Then the 4th one that's kind of harder to remember is a scatter plot. And it's important for you to understand that you don't just use any graph depending on your mood or what you think is prettier to draw. Certain types of data leads to certain graphs being more or less appropriate. And so I want you guys to understand not just what those 4 graphs look like, but when to use them. And so we'll We'll start basic, which is the bar graph. And anytime you're just simply comparing categories, comparing values with between different categories, a bar graph is most appropriate. Examples here would be if you grew different plants and wanted to measure their final heights, if you were graphing the amount of wins of the different NBA teams, that would all make sense to use the bar graph. Here we have the category is the 8 different planets in our solar system. The dependent variable is frequency of ridicule, how often these planets are made fun of. You can see there's a clear winner. Next, we have a line graph. A line graph shows how the value of something changes over time. Notice I bolded over time. That's key here because pretty much any instance that the x-axis is measuring time, a line graph is going to be most appropriate. So here, if you're growing plants, but you actually wanted to track their growth over 2 weeks, a line graph would work. If you were tracking the average temperatures of planet Earth over 10 years, a line graph would work. Here we have time being the age of someone from 0 to 80, and the dependent variable being measured on the y, how often they swear. I agree. Next, we have a pie chart. This shows parts or percentages out of a whole. So when you're dealing with some sort of a data set where there's a known complete amount, such as the percentage of grades amongst my students, well, I know I have 32 students, and so we could make a pie chart there because out of the whole, I could figure out what percentage have an A, B, C, D, and F. Couple of my favorite pie charts. Not sure if you guys are old enough to get this. And last, we have a scatter plot. Here's where we are looking at a relationship or correlation between two distinct variables. Again, often, almost always, independent variable on the x, dependent variable on the y. With a scatter plot, you can look mathematically at the relationship between those x and y variables. How much of an impact does the x have on the y? This might be familiar from math class, but one of the values of a scatter plot is you can look at correlation strength. And so you can see in the upper left here, we see a graph with a clear positive correlation. When the x value is higher, the y value is higher. On the bottom in the middle, we see a negative correlation. When the x is higher, the y is lower, but clearly those variables are still related to one another. Okay, and then the last bit for graphing is this acronym that I think is really important. It is HALT, H-A-U-L-T. And this stands for the 5 aspects of a graph that you need to understand when both you are reading a graph and trying to comprehend it, or when you are making a graph and want to make sure that you include everything that needs to be in there. So H is pretty straightforward. That stands for header. Make sure that the header of a graph is always descriptive. A stands for axis labels. You want to make sure that both the X and Y axes are labeled. U stands for units. Want to make sure that you include units, whether it's the X, Y, or both. L for legend. Not all graphs require a legend, but if a legend is appropriate, it needs to be clearly labeled. And then lastly, I would star this one because here's where students often forget. But T, which stands for trend. Trend is basically the gist of the graph. In other words, what are we learning from this graph? What are the patterns? What are the interesting relationships? And so, again, using this acronym can be beneficial if you see a graph on a worksheet or an exam and you don't understand it. Just go through the levels. First, read the header. Then read the axis labels. Look at the units, the legend, and then try to come up with, what pattern is this graph showing us. And then just as important, whether it's a worksheet or a lab report, but if you're asked to make a graph, I'll often give 5 points for a graph and I'll check off 1 point per letter. Again, a legend is not always necessary, but all graphs you need to include a header, you need to label your axes, include units where applicable, and then lastly, right underneath the graph, you can just write 1 or 2 sentences that tells me the trend of the graph. Again, the overall pattern of the graph, what we are learning from reading that visual. So let's do a quick example. You should be able to look at this graph and clearly read the header, and that tells you what the graph is about. You look at the axis labels and the units, and we see what's being measured on the X, what's being measured on the Y. The legend shows us the difference between the solid bars for grade and the diagonal bars for attendance. Notice that a bar graph is most appropriate here because we're comparing the different categories of students, or the 6 different students. And then take a second here to try to come up with what you would write for the trend. To me, it seems pretty clear, but I want to see if you guys can do it on your own. So go ahead and pause the video, try to come up with the trend. Okay, hopefully you're able to figure out that school attendance is positively correlated with science grade. Students that go to school often tend to do better in school, like Fred or Chuck. Students that don't go to school as often, like Ben and Earl, tend to do worse in science. So I would say that would be the overall gist or the overall pattern of this graph. So that is HAULT, H-A-U-L-T. Okay, that is it for graphing, but we're now going to switch gears into data and measuring, making sure that we understand how to collect data and how to statistically analyze or process data. First, a little bit about the metric system. I hope this is not brand new to you guys, but scientists use the metric system. You might think, well, why don't they use inches or Fahrenheit or pounds? And that's a standard system that very few countries on Earth use. The United States just happens to be one of the few. Because science is a global entity, because scientists collaborate from countries all over the world, it makes much more sense to use the global system than the American system. So the standard measurement for science and most of the world is metric. The units that we use for metric, when you're measuring distance or length, we use the meter. For volume, we use the liter. For mass, we use gram. And for temperature, we use Celsius. I know that standard measurements seem easier, but the metric system is basically based off of tens and hundreds and thousands, and it's actually much easier to use in general. It's just that because the majority of us grew up and learned how to measure things in the United States, we use things like inches and ounces and pounds and Fahrenheit, and we use a different system than what most of the world. The prefixes you don't need to memorize, but it's pretty interesting that just by understanding these 6 Latin roots, you would know how big or small something is. So I already know that a centiliter is 10 times smaller than a deciliter, or that a kilometer is 1,000 times longer than a meter. And a meter is 100 times longer than a centimeter. And this is true with grams. You have kilogram, milligram. It's true with liters, milliliter, kiloliter. But again, it's a pretty useful way to do measuring. And so that's why science uses the metric system. So quick review on data. Data is any factual information that is used for reasoning or for calculation. This is review from last notes, but data can be both qualitative or quantitative. Both can be extremely. Now, statistically, we can put data in 2 different categories. Data that has yet to be processed is considered raw data, whereas data that has been analyzed or statistically processed, we simply call that processed data. And so I'm going to show you guys the different techniques that are required within our lab reports. You don't need to memorize these equations, but you do need to write them down and know how to use them, and then just as important, know when to use them. So the first one would obviously be raw data. Any scientist is going to include their raw data in a data table, in a graph, but in a way that we can clearly see the original values, the original numbers that were recorded during experimentation. But in terms of the data processing, mean, which you guys know how to do, you know that you just add up the values and divide by the total number of data points. Median, which is the middle value of the data set. We very rarely use mode. Pretty straightforward example here. If I have 6 plants and they grew to the following heights, I add them up, I divide by 6, and I get a mean of 25.2. As far as the median, there's no middle value, but the middle values are 24 and 25. Therefore, the median is the mean between those two median values. Okay, next is percent change. This is the difference between initial and final values. What we do to find this information is we take the initial value, we subtract the initial value from that, we divide it by the initial value. And then we multiply by 100% to turn the decimal into a percentage, and then we have percent change. It's really important that you recognize that this is only to analyze values from the same group. So if I had plant A and plant B and I grew them over a span of 2 weeks, I cannot compare plant A to plant B using percent change. I can only look at the growth of plant A from the beginning to the end, or plant B from the beginning to the end using percent change. It's only to look at values within a group and how they've changed over time. Now, if you wanted to compare groups, if you wanted to compare plant A to plant B, you can use percent difference. Often this is used to statistically compare the control group and the experimental group, but any two groups, any two values that are different can be compared using percent difference. So here, you take the difference of the values and divide that by the average of the values. Multiply by 100% to turn the decimal into a percentage, and then you have percent difference. And as I mentioned, this is only to compare different groups, plant A versus plant B. Okay, quick example. If this is easy for you, great. If you have some struggles with it, that's okay, but go ahead and pause the video and try your best to answer both of these questions on your own. Okay, so for the first one, we're going to find the final value, which is 4.4 centimeters, subtract the initial value, divide that by the initial value, and we get negative 0.083. We turn that into a percentage by multiplying by 100%, and we get a percent change of negative 8.3%. For the other experiment, and I'm sorry guys, I did not mention on the previous slide that percent difference, the numerator is absolute value. What that means is that we're always going to have a positive number. Percent change can be negative, but percent difference is always going to be positive. So you put those that equation in absolute value bars. But anyways, to find the percent difference between these two plants, we find the difference of 4.4 and 5, which is 0.6. We then divide that by the average of 4.4 and 5, which is 4.8575. I don't know. But anyways, you divide the two and you get 0.127 multiplied by 100%, and you get 12.7%. I rounded to 13%. So if this was easy for you, great. If you had a little bit of struggle, that's okay. If you're totally lost, that would be an example where maybe you can send me an email. You can send me a Schoology message and we can do some more practice together. But hopefully that wasn't too challenging. All right, and then the last stat that you need to know is standard deviation. This is a measurement of how precise your results are. In other words, it looks at consistency. Theoretically, if you do the same experiment over and over again, it should be fairly similar, and that would give you a low standard deviation. Now, this is important because students often mess this up. This is only to compare trials within a group. So plant A versus plant B would not be standard deviation. Plant A over time, how it's grown over 3 weeks, that would not be standard deviation. The only example of standard deviation would be if I had 5 plant A's that were all given the same treatment, and at the same time I wanted to compare how similar those results were to each other. Then I could use standard deviation. If standard deviation is small, that means that your results are precise. If your standard deviation is large, that means that your results are imprecise or less consistent. How to do standard deviation? You do this, then you do this, then you do this, then you do this, then you do this. I put it in red because, first of all, this isn't math class. I don't want to overwhelm you guys with really complicated stats. And second of all, when you need to do standard deviation, first of all, I'll teach you some shortcuts to use in Excel. And second of all, you can just go to a standard deviation calculator online, plug in your values, and have an online calculator solve it for you. So I don't think there's any benefit of learning this equation. Let's say I had 5 trials in experiment 1 and the plants were that tall. And then I had 5 trials in experiment 2 and the plants were this tall. Which one of these would have a lower standard deviation? Meaning which one of these shows greater precision? So I just use a quick standard deviation calculator, and experiment 1 had a standard deviation of about 1.7, whereas experiment 2 had a standard deviation of 6.8. So clearly experiment 1 shows greater precision. Those results are more similar to each other, whereas experiment 2, those results are much more spread out, showing less precise results. Let's say a robot carried out a study and there was no errors at all and everything, each trial was exactly the same as the others. And let's say that in that experiment, experiment 3, the plants grew to be 27, 27, 27, 27, 27. Does anyone know what the standard deviation would be? It would be 0, because there's 0 deviation within those numbers. Obviously, in the real world, it's very rare that you would ever get a standard deviation of 0. Okay, so that is it for the Unit 1 notes. Obviously, experimental design, graphing, data. This video can be rewatched infinity times, so feel free, if it helps, to rewatch it, especially leading up before a quiz or test. But that is it. All right. If you have questions, please write them down and bring them to next class. Bye.